Hello, this presentation discusses one group of organic macromolecules, nucleic acids. The functions of nucleic acids are very diverse. Some are involved in determining which proteins your cells make, and others are involved in the energy needs of your cells. DNA and RNA, for example, are both related to proteins, but it's DNA that is the molecule of heredity. It consists of a code that determines the sequence of amino acids in a protein. RNA helps in making those particular proteins. The three nucleic acids that are involved with the energy needs of cells include ATP, which is a short-term energy storage molecule, NAD, and FAD. Both of these molecules participate in how a cell makes ATP, the process used. Regardless of their diverse function, all nucleic acids are composed of at least one monomer called a nucleotide. DNA and RNA consists of several nucleotides bonded together in a chain. The picture on the left is a nucleic acid chain consisting of 4 nucleotides, which is very short and doesn't really exist in your cells. Typically, DNA and RNA have hundreds to tens of thousands of nucleotides. Each nucleotide has 3 components: a pentose, which is either ribose or deoxyribose, both carbohydrates, a base that contains nitrogen, and a phosphate group. The pentose in DNA is deoxyribose. The pentose in RNA is ribose. There are 5 nitrogen-containing bases in our bodies: adenine, cytosine, guanine, thymine, and uracil. Uracil is not found in DNA, and thymine is not found in RNA. So these two are kind of exchange one another. Thymine is in DNA, whereas uracil is in RNA. The 2 pentoses that I mentioned, ribose and deoxyribose, you've seen before. They're very similar, only differing by the presence or absence of an oxygen at carbon number 2. So recall that deoxyribose does not have an oxygen bound to carbon 2. This slide shows you the structure of all 5 bases. Notice that some of them have 2 carbon rings like adenine and guanine, whereas others only have 1 ring of carbons like thymine, cytosine, and uracil. Just remember that thymine is only in DNA and uracil is only in RNA. I know I've shown you these structures, but you're not responsible for distinguishing between these 5 bases. I just show them to you so that you can see that they all contain nitrogen. The way in which nucleotides are linked together is through a special kind of bond called a phosphodiester bond, written at the top of the slide. The phosphate group of one nucleotide, the one on the bottom right here, this is all part of the same nucleotide, binds to the pentose of another nucleotide, which is shown up here. So this whole thing is a phosphodiester bond, the bracket that I've shown you in red. DNA is the only nucleic acid that's double-stranded. It consists of the 2 chains of nucleotides that are linked together by hydrogen bonds. These hydrogen bonds are between nitrogenous bases. So to bind together, adenine and thymine need to be aligned properly, and they form two hydrogen bonds between them. So one of these is adenine and one of these is thymine. I'll use a different color. Cytosine and guanine also bond together through three hydrogen bonds. So this one might be guanine, this base right here would be guanine, and this one would be cytosine. And I know this because there are three hydrogen bonds between those bases. We will talk about the structure of DNA in more detail later. For now, you should know that DNA is double-stranded. RNA, on the other hand, is single-stranded. Notice again that I don't have thymine as one of the bases. Instead, I have uracil. I've already mentioned some of the differences between DNA and RNA. For example, DNA is inherited, but RNA is made every time a protein needs to be made. DNA consists of genes that direct the sequence of a protein's primary structure. RNA, we don't say has a gene, we say it's a copy of a gene. DNA is always located in the nucleus, or the mitochondria. But RNA, while it's made, the copy of the gene is made in the nucleus, the copy will leave the nucleus and go out to the cytoplasm where protein synthesis will occur. Now it's kind of interesting that our mitochondria, which are little cell organelles, have DNA. But there is a single chromosome in mitochondria that contains genes that help in ATP synthesis. Now I'll discuss the smaller nucleic acids, starting with ATP. ATP is just one monomer. You can't even call it a polymer. Its structure is diagrammed on the left, and it's really kind of complex. But notice that, first of all, there's a pentose, which is ribose, right here. And then there's a base, which is adenine. That's adenosine. That's where the A comes from, adenosine, those two things together. And then there's three phosphates. So here's a phosphate group, here's a phosphate group, and there's a phosphate group. That's where the T and P come from in the name, triphosphate. So ATP is adenosine triphosphate. It functions as a direct source of energy that cells can use to perform work. It can be stored easily in the cytoplasm, easier than glucose. And the reason it's such a good energy storage molecule is due to the fact that the bonds between the first and the second here, and particularly between the second and third phosphate, is very high energy. It can be broken pretty easily and quickly, and it releases a lot of energy. When both phosphate groups are removed from ATP, what remains is a nucleotide called AMP, cyclic AMP, because what happens is this little ring forms between the phosphate and the pentose. You don't have to really remember that. So it's adenosine monophosphate because you've lost two phosphates, and we often see it written as cyclic AMP or CAMP. The last nucleotide to look at is NADH. This abbreviation stands for a really long name, nicotinamide adenine dinucleotide. You don't need to remember that, write it down or anything, but it does explain a little bit about where the letters come from, NAD and NADH has a hydrogen on it. Its function is to carry electrons that have been harvested from nutrient molecules to the mitochondria. So they take this molecule, NADH, takes electrons from nutrients like glucose to the mitochondria and then transfers them to some machinery that helps in the production of ATP. So NADH is going to be really important to us when we talk about ATP synthesis. That is the end of the nucleic acids lecture. Thank you.